Welcome to Educon. In this tutorial, we'll learn about photosynthesis. So, in earlier tutorial, we discussed about nutrition, types of nutrition in different organisms such as bacteria, other microorganisms, plants, and animals. So, what is photosynthesis? Photosynthesis is process of converting light energy to chemical energy. Light energy to chemical energy. It occurs inside chloroplast on thylakoid membrane. Inside chloroplast on thylakoid membranes. Photosynthesis is responsible for oxygen contents, the air we breathe, and every microorganisms, even plants, even animals, they need oxygen to breathe for survival. So we need oxygen. It can be based on this, the photosynthesis can be oxygenic photosynthesis or unoxygenic photosynthesis. Oxygenic photosynthesis means it releases oxygen into the atmosphere, whereas unoxygenic photosynthesis means it doesn't release any oxygen back into atmosphere. So in photosynthesis, the photosynthesis can occurs in two steps. One is light dependent, another is light independent or Calvin cycle. So now we will discuss about light dependent reaction. As I said before, photosynthesis occurs inside chloroplast on thylakoid membranes. So thylakoid membranes, it can be grana thylakoid or stoma thylakoid. So grana thylakoid will be like this. It appears like this. So on this membrane, the photosynthesis occurs. So inside the chloroplast, it is called lumen, whereas outside it is stroma. So where photosynthesis occurs in plants, it occurs on thylakoid membranes inside chloroplast. Now coming to thylakoid membranes. On thylakoid membrane, we have four major protein complexes like number 1, photosystem 2, number 2, cytochrome B6F, number 3, photosystem 1, and number 4, ATP synthase. So all these four protein complexes are essential in performing light dependent reactions. Let's start how the process begins. Now on my left side, you are seeing an image. This is the thylakoid membrane. So on thylakoid membrane, we have four protein complexes which are photosystem 2, cytochrome B6F, photosystem 1, and ATP synthase. So you might have observed photosystem 1 here and 2 here. Why it is mentioned here? Because based on the discovery, it was discovered first. So at first we thought the reaction starts at photosystem 1, but it was concluded that the reaction first started at photosystem 2. Now let's see what happened when the light hits the reaction center inside photosystem. Electron get excited. This electron will be accepted by a molecule, a modified chlorophyll molecule called pheophytin. This is the primary electron acceptor in photosystem 2. So this after accepting the electron by the pheophytin, it will be transferred to a quinone molecule, which are the membrane transporter. Okay, here the PQ will be reduced to PQH2 because on the left side you might have seen electrochemical gradient for hydrogen ion concentration. these hydrogen ions are lumen side is higher compared to stroma side. So in this side, the hydrogen ion from the stroma side will be reduced, forming PQH2. Again, the electron will be transferred to plastocyanin with the help of cytochrome B6F protein complex. As the electron moves from here, it loses energy. Again, when the electron reaches PS1, photosystem 1, a photon hits again. Again here the electron get excited. This electron with the help of hydrogen ion is used in reducing NADP to NADPH. So our product NADPH is formed in, that is the first product here. Okay, and here once the reactions have, when it loses electron, what happens? It needs electron to compensate for its loss. So two water molecules break down into hydrogen ions and oxygen. This oxygen will be released as a waste product here in the photosystem 2. Okay, now coming to here, now we know the light hits the photosystem 2, the electron excited, the electron flows through electron transport chain involving quinone molecule B6 cytochrome B6F and plastocyanin, and again electron excited with another photon here at photosystem 1. So this appears to be jet scheme. So this is also called jet scheme reaction. Okay, now coming to ATP synthesis. So as you know, the electrochemical gradient, the higher concentration of hydrogen ions on the lumen side will result in the synthesis of ATP from ADP. So hydrogen ion will be accepted here, where it will be where the ADP will be reduced to ATP. So the final outcome in the light dependent reactions are NADPH and ATP. These two compounds are used in Calvin cycle to fix carbon in carbon dioxide, and finally the outcome will be the production of carbohydrates, lipids and proteins. So see, this is a simple explanation for photosynthesis. Thank you.